Okay, so what I'm going to do in the first part of this boot camp is to give you an overview of nanoscale science, and hopefully in the context where it's useful, I guess, for you, for clinicians. In the second part, I will be telling you a little bit about our own research, and I think it fits really nicely with a lot of the ongoing efforts that we have at Northwestern. Okay, so let me just start off with a sense of scale. So when we say nanoscience, I want to calibrate us because nano is a very large range, even though it's a very small length scale. So everything on the right-hand side of the screen, these are all biological entities, all the way from double-stranded DNA, HIV virus, red blood cells, human hair, all the way up to one of the largest self-assembled structures, which is Yao Ming. Okay, he's pretty impressive in terms of this height range here. But similarly, on the left-hand side of the screen are structures and architectures that we have made deliberately at both the nanoscale, see this is about 1 nanometer here, all the way up to the millimeter scale. But the reason I chose this type of image is because it's an exquisite collection of transistors whose gate lengths have now been reduced to about 15 nanometers also. So the ability to really structure things at the 1 to 10 nanometer length scale is really important. And what we'll focus on mostly are these types of materials, because as I'll show you in just a minute, it's the materials that have driven the discoveries. And similar to how tools have always driven new discoveries in biology, that's the same for the advent of nanoscience. So here's a brief overview of some of the key highlights. Okay, you can see that there's a very early period in nano and a latter period in nano. And so in the very earlier period, I would say that people had been taking advantage of the unique properties of nanoscale science without really knowing it yet. So this is an example of a Lycurgus cup, and it was on loan here at the museum. I don't know if anybody had seen it. Okay, no, nobody gets out. But anyway, it was in Chicago, and so you could see this glass cup that has embedded particles in it. And nobody knew that at the time. What they knew is that if you look at the light and reflection, it looks green, but if you put a light bulb inside the middle of the cup, it looks red. And so it's like, wow, it just seems magical. And people said, okay, well, let's just continue to make artisanal quality goods this way. And that's been true for many, many years. Okay, but Faraday had decided that, and this is many years later, okay, 1,400 years later, that he would keep a lab notebook, and this is a really good lab notebook, it's lasted until today, of what he called finely divided gold. And so they have these slides, and he knew something was happening when he put a gold precursor into a solution. He shined light through the solution, and for some reason he could see the light beam, it scattered. And that shouldn't be possible if you just had a solution of, like if you have a solution of methanol, if you shine light on it, nothing happens, right? Nothing is scattering it. So something was going on, and he didn't know what yet. And he couldn't know yet until later, and I'll show you when that could happen. So part of the key time happened in 1959, and this is when Richard Feynman gave his famous speech. And he said, it's called There's Plenty of Room at the Bottom, an invitation to enter a new field of science. And I'll just read it to you. So people tell me about miniaturization, how far it has progressed today. They tell me about electric motors that are the size of the nail on your small finger. And there is a device on the market, they tell me, by which you can write the Lord's Prayer on the head of a pen. But that's nothing. That's the most primitive, halting step in the direction I intend to discuss. It is a staggeringly small world that is below. And in the year 2000, when they look back at this age, they will wonder why it was not until the year 1960 that anybody began seriously to move in this direction. So his ultimate question was, why cannot we write the entire 24 volumes of the Encyclopedia Britannica on the head of a pen? Okay, and so at that time, okay, they didn't have Wikipedia and all of these things, so you have a finite amount of knowledge, and if you take that finite amount of knowledge and you were actually to put it on the head of a pen, you would need to be able to control matter at the 10 nanometer scale. And that's not trivial. But the interesting thing is that is the biological length scale, and so new tools are needed. Okay, so what are the two new tools that enabled us to resolve things at the 10 nanometer scale? And so I put this date here, which is 1986, won the Nobel Prize. So there were two Nobel Prizes that year. One was for the discovery of scanning tunneling microscopy. So it's the ability to manipulate individual atoms on a surface. And so that's what this is, as well as the transmission electron microscope. And the transmission electron microscope can reveal the size of nanoparticles in a glass. And so it turns out that this is what nanoparticle is from a sliver of this Lycurgus cup. Okay, don't ask me how I know how you broke off a piece of the Lycurgus cup. That's a different story. But this is what it looks like. So this is made of all of these tiny nanoparticles. Okay, in 1996, the Nobel Prize was given for the discovery of Buckminster fullerene C60, another nanoscale material. And in the year 2000, which I've shown here, is another important milestone year, because that's when we had dedicated funds to actually pursue this study. And so in his State of the Union address, Bill Clinton said, "Soon researchers will bring us devices that can translate foreign languages as fast as you could talk, materials 10 times stronger than steel at a fraction of the weight, and this is unbelievable to me, molecular computers the size of a teardrop with the power of today's fastest supercomputers." Okay, so you can see what he was thinking about in the year 2000. We are interested in information processing, we still are. We're interested in superior materials, and we're also interested in the idea of faster electronics. So it was really a great foresight and the advisors that he had with him to say, look, this is something that's really worth investing in. Okay, so after the investment by the United States government for science, the Nobel Prize was also given for giant magnetoresistance, materials-based phenomenon. And then finally, there's these new prizes that came out called the Kavli prizes. So the Kavli prizes are sort of an upstart prize, sort of equivalent to the Nobel Prize in the sense that they're interested in recognizing major achievements that don't have the long track record yet, but that are anticipated to have major achievements. So these prizes are given in astrophysics, neuroscience, as well as nanoscience. And so in nanoscience, the prize was given for semiconducting quantum dots and carbon nanotubes. Okay? So the point of this history is to mostly show you that nanoscale materials has really driven nanoscience. And it's the nanomaterials today that you'll hear about both in my talk and in the subsequent talks that allowed us to do really interesting things in terms of diagnostics and therapeutics. But let me just give you a length scale on the different types of nanoscale materials that are being used today. And this is on a linear length scale. So this is a gold nanoshell. It's really, really big. Okay, this is a carbon nanotube, about 1 nanometer in size. A gold nanoparticle, on average, it's about 13 nanometers in size. And semiconducting nanocrystals, which are about 2 nanometers in size. So you can see that these length scales are perfect for different types of applications, both in vivo and ex vivo. Okay, so the idea that nanoparticles can be used for different types of applications have been around for a long time, whether it's in terms of diagnostics, whether they can be used as delivery agents or therapeutic agents. And again, that's not surprising based on these length scales that we see here. So the questions are, and as we're trying to design new materials for biomedical applications, are what types of materials should we use? They obviously need to be biocompatible, but do they also need to be biodegradable? Does that matter or not? Or can they be cleared from the system? Secondly, we also need to understand a little bit about where we're going to load the drug or where we're going to load different types of targeting ligands. So is the better design to encapsulate it in the core? So for example, should we load all the drugs in the interior of some type of delivery agent? Or is it better to attach it to the surface? So in this cartoon, what you have is the drugs localized in the middle of the core, but then you have these antibody targeting ligands as well as polyethylene glycol molecules to increase the circulation time of these nanoconstructs in the body. And so there's a big range of things that you need to consider if you're actually going to use nanoscale materials in any of these applications, I would argue. But in the beginning, what I would like to do is just give you a short overview of how nanoparticles can be used in diagnostics. And then I'll tell you a little bit about how they're being employed as drug delivery agents as well as therapeutic agents. And I go back to this, which is like almost a cartoon at this point, because it really is the fundamental science that has driven all of the really exquisite applications that you'll see, especially in the talks after mine. And so this was pioneered in the lab of Chad Mirkin, where he said, okay, if I have these gold nanoparticles and I'm interested in putting DNA on the outside. And so if these particles are isolated, the solution just looks red, similar to the Lycurgus cup, it just looks red. But if I can go after a target molecule, and in this case it's just a DNA linker with different sequences, A prime and B prime, and if I add this to a mixture of these A and B nanoparticles, I can get them to cluster together. And by clustering together, instead of this nice red color, they form this blue color. So it's a calorimetric indicator that, oh, you've detected your target. It's very, very simple, and it's very sensitive, and you can tell this by the DNA melting curve. So this was done probably 10 years ago or longer, I guess, at this point. But the emphasis is that it's the fundamental science that has driven these new types of discoveries. So for example, you'll hear later today about these diagnostic designer nanoparticles. So instead of something that just does things in a beaker, you can also start to look at targets intracellularly. And so this time the design is far more sophisticated, but without the fundamentals, you could never get there. And so you have an antisense DNA. This is the recognition sequence, which is covalently bound to the gold nanoparticle. And you also have reporter flare, so you have a dye. And so there's a Watson-base-Crick pairing between this antisense DNA as well as the flare. Okay. And so as soon as these are internalized within cells, you're looking for a target mRNA. And when the particle encounters this target will displace this reporter flare, and as the reporter flare leaves the surface of the particle, then you can actually start to see the fluorescence. So this is a pretty big advantage for being able to detect small sequences of mRNA. And the nice thing, of course, is that you can imagine doing this with multiplex detection. And you can also use this type of system with live cell technologies. So that's another thing that's really important when we're thinking about nanoscience, is that the new technology has to be able to be integrated with certain types of current technologies, otherwise they can never receive widespread use. Okay, so what about therapeutics or delivery agents? What is necessary in the design of nanoparticles? So there's this great review article by Scott McNeil. He's one of the key people that's involved in the nanoscale characterization laboratory. And so what their job is to analyze all of these different types of nanoconstructs that potentially might see some benefit in the market, as well as for future clinical trials. And so these are three things that are important in the design of nanoparticles, because you can't just make random things and have it work. So the zeta potential, or the charge on the particle, is really important. If the charge is positive, you can induce cytotoxicity, which is bad. If it's too negative, that could also be bad. In terms of sizes, you don't want it too small, otherwise it gets cleared by the kidneys. And if it's still of certain sizes, it can also be taken up by the liver. But then again, you don't want it too large either, because if it's too large, even if it can make it into the tumor via this enhanced permeability and retention effect, it can't actually make its way into the cells. So there's a trade-off on all of these different types of parameters. And then finally, you don't want it too hydrophobic, because if it's too hydrophobic, the MPS system will take over, and all the phagocytes will just remove everything. And so then your particle or your agent can actually get to where you want it to go. And so you really need something in this middle region. And what's nice is that it gives us some design rules for how you might want to think about nanoparticle design. Okay, so if you're thinking about agents, especially in vivo, here are some of the principles that have been distilled from all of their testing, as well as from things that are being considered now in phase 2, I think it's phase 2 clinical trials. So you want diameters that are on the order of 50 nanometers or so. Because if it's too small, as I mentioned, it gets cleared by the kidneys. If it's too large, they can't be endocytosed readily. And you really want them about 40 to 50 nanometers, because it's optimal for membrane wrapping. So any type of endocytosis processes, depending upon the particle size, 40 to 50 nanometers is a good size. Secondly, you want the surface to be neutral or slightly negatively charged, so you don't introduce any unwanted opsonization effects. And if you have to choose between larger and smaller particles, the smaller particles are a little bit better, so they're just not scavenged by all the macrophage. So these are some of the design rules that have come up that have informed different types of materials that are currently being tested. Okay, so in terms of drug release, I just want to give you a little bit of an overview on the types of materials and carriers that can be used in biomedicine. Because a lot of the things that you'll hear today just are based on gold nanoparticles, but that's not the only game in town. It's just the one that we're really good at Northwestern. But to give you a broad overview, to be fair, I wanted to introduce these other materials also. Okay, so one of the most popular ones for drug delivery are liposomes. And so in terms of liposomes, you can control their sizes to a range that you might care about. But how are you going to deliver the drug? And so people have spent a long time encapsulating doxorubicin into a liposome, and they've also been able to integrate different types of peptides into the bilayer. And so at a certain temperature, this peptide is closed, but if you just heat the solution or trying to get to physiological temperatures, the peptide can open and the doxorubicin can sort of leak out. So that's one way to have external heating being able to do drug release. Another way is to use a different type of drug delivery agent, which is a porous silica nanoparticle. So in this case, you have silicon dioxide and you have pores in between there. And you functionalize this with DNA. What you then can do is you can introduce drug, which are loaded inside these little pores. And then it's capped with a magnetic nanoparticle with DNA that hybridizes to the DNA that's on the particle. And so in this case, you basically plugged up all of the drug inside this mesoscale silica particle. What you can then do is you can apply an alternating magnetic field. It's at this type of frequency, which produces enough heat that the DNA can dehybridize, which can then result in release of the drug. So you can imagine there are a lot of different strategies that can be evolved for external triggering of drug release. Another example is to take advantage of light and also pH. So going back to liposomes, because this is one of the more popular delivery agents today, what you can do is the pH of within an endosome is lower than externally to the cell, so that is one way that people have been exploring to be able to result in having an external trigger to release drugs. And so if I have a PEG-coated layer around a liposome, if it's sensitive to pH, the PEG can cleave out. And then what's exposed is this TAT peptide, which is a cell-penetrating peptide. So if nanoparticles have this on the outside, they have a much higher probability of being internalized within the cell. So it's a really nice way to be able to get the particles where you want them and internalized. Okay, and so the final, not the final carrier, but one of the carriers that we're most interested in are gold nanoparticles. So there's this one particle, which is a silica core surrounded by a gold shell. These are these gold nanoshells that I told you about earlier. These are also, I think, in clinical trials right now for head and neck cancers. And so the idea is you don't really have ligands on the outside or drugs that are doing anything, but this entity itself is heating. So you shine your infrared light on this particle, and this particle heats the local environment around it to induce cell death, and it seems to be working pretty well. The challenge, of course, is the time that you need to apply for heating is on the order of minutes. It's around 5 to 6 minutes of continuous irradiation, and it's fairly localized. But of course, you can also apply this light-based heating technique to release drugs. So in this cartoon, this is a nanorod. It also has PEG on the outside. You have DNA and DNA that has also trapped different drug agents. And so by shining near-infrared light on this guy heats up, it dehybridizes the DNA, and then the drug is released. So what I wanted to communicate in all these different strategies is that you need control over the particle, and you also need control over the ligand. So there's a lot of chemistry and engineering that has to be involved in the success of these types of structures. Okay, and so that's sort of a general introduction to the types of problems and possibilities that we and others are interested in. So we have 3 talks, and I'll be giving the first one. And then we have one on nanoflares and the one on RNAi-based nanotherapeutics. But before we transition to the more technical talks, I'd be happy to answer any questions if you had any at this point. Yeah. I'm sorry, can you repeat that?
How are they metabolized?
How are they metabolized? The liposomes? Yeah, so they're mostly made of these lipid bilayers, right, that are surrounding some sort of core. I think that they're mostly, so they're not taken up by the MPS that much. I'm not sure about the actual mechanism of the degradation, but since they're soft, you can imagine that something can potentially insert into the membrane and for disruption, but I'm not really sure what the detailed metabolic pathway is.
Do all cells have targeting molecules? And if not, can you create them?
Do we need targeting molecules? Okay, that's a good question. So I think there's a first order and a second order effect. So most of the internalization of the nanoparticles in tumors has been by this enhanced permeability and retention effect. So the vasculature of the tumors are just larger, and so the nanoparticles that have been loaded with drug, they're already accumulating there. But there is an enhanced effect if you are able to target the biomarkers that are expressed on the tumor surface. But I think that the primary effect is the EPR. But the reason why people are interested, at least going to through FDA approval in targeting, is because you don't want unwanted accumulation in all of these other organs. So all of these other organs are going to be able to take up nanoparticles, but if you have a place where they're targeted, then the hope is that all of the nanoparticles will go to based on the targeting to the tumor and not be distributed in unwanted organs. Yeah.
So I heard you mention about screening for safety and for screening the safety and ensuring that these things are not going to cause damage and detrimental effects. I didn't hear anything about immunogenicity. The molecules of the whether it's RNA, DNA, or proteins attached to the surface, are the studies being done to look at how the immune system senses this?
They are. So that's one of the big questions: is what is the immune response? What is the response in the presence of all of these foreign agents? And it's actually a subtle question, because sometimes you get an elevated immune response, but it doesn't produce adverse effects. It's just that it's an elevated response. And so what most people are doing now when they're trying to analyze the therapeutic applications is they're working with immunologists at the same time to actually assess both just the therapeutic effect as well as the immune response. But I know by talking to people that have taken drugs to phase 2, just because you have an immune response doesn't mean that it's adverse necessarily for the effectiveness of the particle. But it's a little subtle point, but it's a good question. Okay, so now I'll transition to tell you a little bit about our own work, and I'm a big fan of gold nanoparticles, and we at Northwestern are a big fan of gold nanoparticles, so you hear a lot about that today. But I want to tell you a little bit a story where this is the drug delivery agent, but also they're hitching a ride on some type of protein that can actively traffic cargo. And the reason we're interested in this is because what I didn't tell you earlier is that when the nanoparticles are internalized, they're just sort of floating around, okay? So you want them to go to specific locations. You don't want them to just be internalized and hopefully they make their way to an organelle to do its business. And so by being able for them to hitch a ride on a protein that has shuttling properties, there could be some promise in this. Okay, but in our case, we're using these gold nanoparticles as drug delivery agents, but we also want to have the opportunity to release the drugs from the surface if we want to. And so in that case, we need to design particles that have an optical response in this near-infrared window, or these optically transparent window. So if the wavelengths are too short, it's going to do DNA damage, but also the light is not going to be responsive to all of these elevated levels of oxygenated hemoglobin and other proteins that are in the system. So we're interested in designing particles with an optical response in this near-infrared window. Okay, so I told you earlier that according to the design rules that have been proposed, you want particles on the order of 50 nanometers or so, 30 to 50 nanometers in size. But in order to have them absorb at near-infrared wavelengths, you can't use spheres. Because if you had spheres that were this size, they would just absorb green light, and green light is not good, right? If I take this laser pointer and I shine it on me, it's not going to penetrate through the skin very much. So in order to have this type of size, but also to achieve absorption at these wavelengths, we have to have anisotropic shapes. And so this is the star shape that we have decided to investigate. In addition, we want these particles to be slightly negatively charged. And so what we've done in our case is we've chosen oligonucleotide ligands, and in this case it's a DNA aptamer, because the backbone, the phosphate backbone, makes everything fairly negatively charged, but not too negative. Okay, so this is the bare minimum of what we need to accomplish. But as a physical scientist, we're interested in designing things with optimized properties. And so we have sort of a dream list of things that we'd like. So the first thing is that we would like to have cell surface targeting ligands that can also act as anti-cancer agents. So we're not interested in making what I call Christmas tree molecules, okay? Christmas tree nanoparticles. I don't want a nanoparticle with 10 to 20 different functionalities, because more than likely it's going to be difficult to achieve to scale that, but also to have uniformity when you're trying to go through FDA approval. So the idea is, can we make things as simple as possible? And one of the simplest way to do this is to have the targeting ligand also be the agent. Another thing that we're interested in is whether we can get on-demand release of the drugs. So we'd like the particle to end up where we want to intracellularly, and then have an external trigger so that the drugs can be released. We don't want just somehow the particles to be taken up and then randomly the drug is released. We'd like to be able to do that on demand. And then finally, we'd like to be able to achieve organelle-specific delivery. Okay, so which is ambitious. I should mention that most of the work right now is on just cell surface targeting. If you can just achieve cell surface targeting, that's a big benefit. But we can always dream as scientists, and so if we can get organelle-specific delivery, then our hypothesis is that the treatment will become more efficacious. And so we've done several things to try to go along these goals. So we're using a DNA aptamer that acts as a cell surface targeting ligand as well as an anti-cancer agent. So this particular DNA aptamer does both. In order to get on-demand release, we're still using light pulses, okay, similar to this near-infrared range, but instead of continuously having to shine light on the particles, we use these ultra-fast femtosecond pulses. So femtosecond lasers have started to be incorporated in a wide range of biomedical applications, especially for LASIK. And so in terms of organelle-specific delivery, why not? We might as well just target the nucleus. There's only a couple organelles that you'd probably be interested in. One is the nucleus, and the other is probably mitochondria. Okay, so this is what we're interested in doing. So this is our concept. So it's a new concept from our perspective, but from a fundamental level, we're interested in how nanoparticles interact with cells, okay? And they interact with cells at very different length scales, and so we need to employ a lot of different tools to look at everything in a step-by-step manner. So this is the nanostar. This is a cartoon, and this is the DNA aptamer, AS1411. So it was in phase 2 clinical trials for renal cancers as well as acute myeloid leukemia. So this is not something we invented, but it's something that's just that's in the literature. So this is our drug-loaded construct. What we're then interested in is taking advantage of a general biomarker that's overexpressed on many different cancer cell surfaces and have this guy bind to the general biomarker. The third thing is we'd like to be able to actively transport the construct once it's been targeted to the proper cell type and have it be delivered where we want. And then finally, we'd like to be able to release the drug molecules on demand. So the reason that this is important is because, for example, say the cells are internalized here. I don't want to release the molecules at this point because the molecules, at least the action of the molecule, is not effective until it's at the nucleus. Okay, so there's a reason why you might want to have on-demand drug release. Okay, so this is the protein that we're interested in. This is the shuttle protein. It's called nucleolin. It's 105 kilodaltons. So in normal cells, it's abundant in the nucleolus, and it's involved in ribosome biogenesis and regulating cell growth. It turns out it has a range of different functions, and so all you have to do is disrupt one of its functions for it to be useful as a cancer target. What it also does is it shuttles between the nucleus, transport cargo between the nucleus and the cytoplasm via the nuclear pore complex. And in normal cells, it also stabilizes anti-apoptotic Bcl-2 mRNA. But in cancer cells, it's overexpressed in the cytoplasm, and it's also overexpressed on the nucleus. So if we can take advantage of its, if it's cancer, if its properties in cancer cells, where it's overexpressed on the cell surface, as well as part of its normal functions in terms of shuttling between the nucleus and cytoplasm, we might have something really special. Okay, and I should mention that this is a ubiquitous protein. And so this is what the structure of the aptamer looks like. It's a short DNA oligomer. And a definition of an aptamer is that you're interested in binding to a protein or any other type of target by changing shape. Okay, so it's, you can think of them as synthetic antibodies, but they're much, much smaller. And so in this case, the shape that it forms is this homodimer. So this is one strand of the AS1411, and this is the other strand. And when it forms this homodimer, it forms these G-quartet planes. And it's these G-quartet planes which it causes or endows stability to this drug molecule. That's why people have been interested, not just in this molecule, but in the ability to design materials with these G-quartet planes for stability. Because one of the main problems of free DNA or free RNA, siRNA, is that it almost immediately degrades if you just use it in its free form. So people are trying to figure out ways to stabilize it, whether it's either on the particle or through something like this. Okay, so the use of free aptamer, this AS1411, has been tested in vitro for some breast cells, and these are the normal cells. And what they're doing is at micromolar concentrations, so very high concentrations of free drug, they're continuously treating the cells. And so you can see after a certain period of time, the cells don't continue to grow anymore. They don't divide. They all end up dying. While in the normal cells, they continue to proliferate, and it seems to be fine. There was this recent study that just came out a couple of months ago on the use of this aptamer for invasive breast cancer and the downregulation of certain microRNA. So nucleolin does just sort of a lot of different things. And I'm mostly going to talk about this later for some of the work that we focused on. Okay, so how do we make our particles? So as I mentioned, we want to have a very simple design because if we have a simple design, then ultimately it can be simply scaled. We also wanted it to be biocompatible. And so all we're doing is using this goods buffer, which might be familiar to many of you in cell culture and media. And then we just add it to this gold salt precursor, and out pops nanostars. It's like really, really easy. It's like a room temperature reaction. Okay, and so we can make a lot of it. And so this is a liter of what some of the nanostars look like. This is a transmission electron microscope image of them, just so you can see how electron dense they are. but here are some higher resolution images of these guys. So they tend to have, 5, 6, 7 branches. Sometimes they can be longer than others. You can actually control the length and the ratio of these branches by changing the relative concentrations of these guys, as well as by changing the pH at which we do the reaction. Okay, so then what we need to do once we have these stars is that we want to test its absorption properties, and so this is good news for us. So it falls within this near-infrared window between the 650 and 850 nanometers. And if we use dynamic light scattering to assess roughly what the size is, the average diameter is about 25 nanometers. I should mention that this is not a perfect way to assess size because it's anisotropic in shape, but it's sort of good enough to give us some ballpark value. Okay, what we then want to do is we want to put the aptamer onto the surface, and part of the reason why all of us here at Northwestern like gold is because gold is very easy to functionalize through a thiolate bond. And so you can put a thiol onto any type of DNA or siRNA, and you can, under the appropriate reaction conditions, you can just form a chemical bond. So once you form a chemical bond, this type of entity is extremely stable. And so we also do dynamic light scattering to make sure that we have the relative increase that we would expect if we actually did attach the drug molecules to the surface. So then the question that we were interested in asking was, as we were starting the in vitro work, was how long do we need to incubate these particles with the cells so that they get to the nucleus? We have no idea how long that would take. And so what we ended up doing is we took the aptamer and we labeled it with a Psi5 dye. And so we incubated for 2.5 hours, 5 hours, 7 hours, 24 hours, and this is the control. And the blue is the DAPI-stained nucleus. And so it's a little bit difficult to see in this light, but by the time you get to 7 hours, for sure 24 hours, all of the red signal is overlapping the stained nucleus. We feel like, okay, 7 hours should be enough for a large population of the nanoparticles to end up at the nucleus. But of course, confocal microscopy can't really tell you any nanoscale information, right? It's just a two-dimensional slice of what's happening. So in order for us to verify this, we need to use transmission electron microscopy. And so I really like these images because what we're interested in is saying, okay, you have these nanoparticles, and at this position, they've started to form these little clusters when they're at the cell nucleus. But you're able to see the particles indicated by these black arrows and these dark spots accumulate around the nucleus. But what's more interesting than that is as these particles are at the nucleus, you see that these invaginations. Let me see if I can point this out like this. So instead of a smooth nuclei like this, you have these points that go in. They're sort of indicated by the white arrows. So what's really interesting about this is the first time that it's been reported that you could actually see what this AS1411 drug does, okay? Because it's a molecule, right? You can't see the DNA in any type of way. Fluorescence is not going to give you enough information resolution-wise, but because you've attached it to the gold, and in this case, the gold acts like a marker. The gold marker says, okay, here is the marker, and I have drug on me. And so what you can see is that the molecule is actually producing all of these invaginations. It's a one-to-one correlation. So it's pretty neat. It's the first time that there is an observed associated physical change of the drug interacting with a specific organelle. Okay, and the other thing that's interesting is that the nanoparticles don't make their way into the nucleus. The nuclear envelope just folds all the way around it. So we did control experiments with the control aptamer, and this is all in HeLa cells as a model system. And it was really difficult to find any particles near the nucleus in the control case because it doesn't bind to nucleolin, and so nucleolin does not shuttle it to the nucleus. But you can see there's a big difference in shape here and then with the drug-treated nanoparticle. The other control that we looked at was another epithelial normal breast cell, and you can see that the nuclear shape looks really nice and there's not any types of deformations. So we also did the control experiment where if you just dump a whole bunch of micromolar concentrations of drug, you can indeed see the invaginations. And so this is an effect of, and potentially could affect the mechanism of how the drug works. Okay, but then what we were interested in is, are there any adverse effects if we tried to release the drug? So in this case, the drug is trapped on the nanoparticle surface. And so what we did is we ended up shining near infrared light, but we only want to shine light for 2 seconds, okay? We'd rather not shine light for 5 minutes, which is a long time. But if we shine light for 2 seconds and we use femtosecond pulses, we can actually break chemical bonds. So we break the gold-sulfur bond, which then releases the drug molecule from the surface. And so we did flow cytometry to make sure that we were actually releasing the dye-labeled drug molecule, as well as we assessed whether there was any adverse effects by shining light. And it turns out there is not. So 2 seconds is not very long. Okay, so what happens when you actually shine light on the drug? Does that change anything in the way that the action of the construct? So this is a different nuclei, and you can see the nanoparticles here, and you can see one invagination here, another invagination here. But if you release the drug molecules from the nanoparticle, the deformations are exacerbated a lot, okay? So instead of just a few that correspond mostly to where the location of the nanoparticle is, all of a sudden, instead of something that looks like this, you have something that has, massive folding. Major changes in phenotype. So this is pretty interesting to be able to start to say, okay, you can already make something better by putting it on a nanoparticle, and then you can even make it better if you can actually release the drug at the site where you might want the action to occur. Okay, so then we carried out some assays to make sure that, to see whether there was any correlation with these changes in phenotype with the biological activity. And so we carried out this late-stage apoptosis assay for just the nanoparticle as well as the nanoparticle, and we released the drug from the particle. And so you see an increase in late-stage apoptosis, so that's positive. What we also did is to look at double-stranded DNA breaks within the cell nucleus. So this provides some preliminary evidence that the released drug is actually making its way into the nucleus. So for example, in this case, we're using an antibody stain for these double-stranded breaks. It's binding to certain aspects on the histones and the chromatin structure. There's nothing in the nuclei, it's just blue. But in this case, you can see the signatures of these double-stranded DNA breaks, and that can only happen if this AS1411 molecule has actually threaded its way through and inside the nucleus. So I think this is pretty exciting. Okay, finally, we want to look at population effects. So not on the single particle level, but how are things acting on a whole system. And so we did all these control experiments with the light released, with not, with the control aptamer and the light released with not, with these epithelial cells, as well as just the free, 10 times the amount of free drug. And so let me just compare these guys here. So just look at the effect of you're releasing the free drug, and then you have massive concentrations of the free drug. So I should also mention in this type of graph, in this type of plot, we're only shining light on it once, okay? It's not continuous irradiation. So this is just 1 2-second irradiation, and then you're looking at the effects immediately after radiation, 24 hours, 48 hours, and 72 hours later. So what's interesting, if you look at, say, 48 hours after release, you can see that the viability or the cell death has increased to about 60% if you compare the drug release from this guy compared to the free aptamer, which is 10 times the amount of concentration when there's been very little cell death. And so what this means is, I'm sorry this is a lot of text here, is that once you release the aptamer from the concentrations that we're using for treatment, if you released 100%, it would be about 33 nanomoles, which is very low. And this 33 nanomoles, which is near the nucleus, gives you far better effects when you're comparing at least the cell viability than 450 nanomolars of freely diffusing AS4111 in the cytoplasm. Okay, so by being able to both target the organelle and to release high concentrations at the site of the organelle, the efficacy is vastly improved. So I think this is pretty exciting preliminary results. Okay, and then the other question they were interested in is HeLa cells are just a model system. Everybody likes them because they're easy to grow. But can we take advantage of nucleolin as a ubiquitous biomarker for other types of cancers? And so what we did is we did this, we tested the system in a cancer panel of 12 cancer cells, a range of different types, and then we also have a normal cell. We also did, it's hard to see in the confocal images, but we also did this Western blot analysis to categorize how much nucleolin is also on the surface as well as in the nucleus and cytoplasm. And all of these guys have overexpressed nucleolin. And so the first thing that we were interested in testing is the reason why AS1411 works is because it's competing with the stabilization of nucleolin with its anti-apoptotic gene. Okay, so if we can destabilize the interaction between nucleolin and this Bcl-2, then using the AS1411 to bind to nucleolin, then you're going to get cell death. It's this, so basically you're inducing apoptosis. That's the mechanism for how this works. So if we look at Bcl-2 mRNA expression, you can see for all cases, if you look at the red hatched, the expression is reduced far more with the release-free drug compared to just 10 times the concentration of the drug that's added. And what's nice is that it works for all different types of systems. So we've not only confirmed the mechanism that has been shown that works for the free drug, but we've also been able to show that when you have the drug on the particle and then you can release the local concentration of the drug, that the response is even better. And then similarly, we did this viability study, again, across this whole range of cancer cell lines. And on average, what you see, this is sort of a lot of work. I mean, I'm a physical scientist, so we're used to working hard at experiments, but biology is a whole different beast. We're doing like 10 times the amount of experiments that we ever did to confirm what we already thought. But I guess it's more satisfying that way. I should say it like that. And so this is a lot of work to put all this together, but on average, there's about a 70% decrease of cell viability after you release the aptamer. And you only get about less than 10% with the free at 10 times the concentration. So we're always comparing to the free aptamer because you want to make the case that the nanoparticle is doing something better. Okay, and then I'll just finish up with where we're going in terms of scalability. So this is what we can do. This is, say, 5 liters of nanoparticles. And then you can centrifuge it down and you can concentrate it 100 times, and it basically looks like this. This is the leftover. It's like a sludge. It's really, really thick. But the people in the developmental therapeutics core can still draw it up into the syringe, so that still works for the biodistribution studies. And so we were looking at toxicity at these concentrations, 48 mg per kg, 9.6, and 4.8. I think we actually have 6 of them, 6 different ranges that we're interested in. So this is pretty high. Most of the time you don't have nanoparticles at this high concentration. So we did some blood work, and basically we have 3 female rats, 3 male rats, and you can see that at least within these green bars, the green is the acceptable level. So all these concentrations of these particles that we looked at in the rats, they're all within this acceptable level for hemoglobin levels, red blood cell count, and the platelet count, and these other types of characteristics. This is after 2 days. Similarly, after 14 days, it also, if you look at these green bars, it also looks good. So you still have continuous circulation in these systems. It turns out the animals don't pee these out at all. Okay, they all stay internalized. And so we're looking at the liver. The liver is healthy after 14 days, so this is the control, and this is what it looks like at the highest concentration that we're using. And so they're always found in these macrophages, so that's not too surprising if they end up in the liver, and they sort of look like this in these different clusters here. The spleen is also healthy after 14 days, and this is 100 times the concentration, and you see these relative cell phenotypes, but they're much less concentrated compared to the liver. We also did histology for this type of system, and we don't know how to interpret these, but our experts tell us that the structure looks normal for both the liver at all of these concentrations, as well as the spleen, and that there's no abnormal lesions or signs of pathological processes. So we're very confident in these types of results. So what that means is we do all of this work to show that they're not toxic, cytotoxic. So I guess that's a good thing. It's just from my perspective, it involves a lot of animals, more than you would have expected. Okay, so in summary, let me summarize what I've tried to tell you. I've introduced this multifunctional, multi-scale nanoparticle. You can do a lot of things to control the optical properties. You get these near-field enhancements at the tips. That's where the drug is being released. And you have this plasmon resonance that is tunable by changing shape. I only talked about this today, but it turns out shapes like this are producing really interesting results when you load MRI contrast agents on them. We have loaded oligonucleotides. You get higher loading. They're very stable on this type of structure. And this is the first time that you've been able to load homodimers in this type of way. I think this has possibilities as a new formulation of approved drugs. So if there's other types of aptamers, and we're looking at these also, you can load them onto the nanoparticles, and they're stabilized, and they act like new entities. But then also, you can achieve multiple functions for diagnostics and therapeutics with these highly stabilized nanoconstructs for neurotoxicity effects in vivo. Okay, and so this is the group that did the work. Mostly, it's the work of Kayla here, who did a lot of the TEM imaging, as well as Duncan, who was also a Malkin Scholar. He was awarded a Malkin Scholar from the Cancer Center for a lot of the work that he really pioneered in this gold nanostar area. So I'll stop there, and I think I have a couple minutes for questions. Yeah.
Very impressive stuff. The fact these things are still hanging around after 14 days, presumably they're going to lose the drug that they're carrying as you release it due to the light pulsing of the. Are they just going to continue accumulating, and if the cell that's carrying them dies, they're taken up by another cell and continue to accumulate, or is there an eventual excretion of these things out of the cells?
Yeah, that's a good question, and this is what we all want to know. Like, what happens to them, right? Or do they just, well, say they get just taken up in the liver, say they just stay there. Is that a bad thing? I don't know. It might be okay. It depends on how you think about implants. I guess it depends on your prescriptive implants. I mean, people have implanted things all the time, and it seems to be fine. But maybe in a nanoparticle case, you might not want that. And so, in terms of your first question, and it will lose its abilities to be an agent. We don't know if the drug is falling off, but we suspect that it is not, even after 14 days. Because it's a covalent bond, which is about 2.5, 3 electron. I mean, it's very strong, and so unless there's a reason for either the DNA to be degraded as it's on the particle, we don't see any reason for it to lose some efficacy. Because the light does not shine continuously; the light must only shine at a certain time. But in terms of the fate, this is what we all want to know.
In the global scheme of things, it looked to me like your lowest viability of about 25%. That's not a terribly low viability. Are you concerned that you have not moved it below that level?
That's a good question. So the lowest viability is like 25%, right? You want to get to like 0. I guess in our defense, what I should say is that this is only for one exposure. So we could potentially expose multiple times for the system. We had just not tested that because it was, we will, but it was too complicated to figure out what's really going on in the beginning. What we do know is getting down to that 25% is that the drug is still working even after 24, 48 hours. And the question is why does it still do that? Because the doubling time for cells is on the order of 24 hours. And so how do you keep getting decreased viability? So we think in terms of population, they must be doing DNA damage somehow. And so as the cells are splitting, they're taking this damaged DNA so that ultimately the global population dies, or it has lower cell death. But we are definitely trying to increase possibilities to either shine light multiple times, or the other way that we found that even works better is to have higher drug loading. So we found that with higher drug loading on the particle, we can already get less viability than we have now. Yeah. Oh, one last question, right?
You did your biodistribution experiments. In those experiments, were they mice xenografted? Or if you have done future experiments, how much of the injected dose actually makes it to the tumor?
That's right. Okay, so the stuff that I showed you for all of the 48 mg per kg and all that stuff, that's all in rats. So that's just rats, no tumor. So we have done things with mice where we have a xenograft implanted tumor. And we tried this with implanted MDA MB231. And so it looked like there was a pretty good accumulation in the tumor, but we were still losing a lot to liver and spleen. I think you will always lose to that. But I should say that that was on an unoptimized particle. And so what we want to do now is create the best optimized particle and redo that test. Because given that these things involve a lot of animals, I don't feel good about just making small improvements to the construct and then testing it. But yeah, that's a major emphasis of what we're looking for. Okay, well, thank you.